Okay. Good morning or good afternoon or noon. Thank you for inviting me. I'll speak about nanoarchitecture of snare protein and clusters. And this is part of a bigger project which we now started to create some kind of a blueprint for synaptic plasticity. I'll try to zoom in from the level of the brain into the level of a single molecule, which will fit, I think, more the audience here. And if we zoom into the brain, in between billions of neurons, we can detect a specific site, which is called the synapse. These synapses are the connecting structure in the cell, in the brain. And this connects two neurons. So if we zoom into the synapse, what we have is many vesicles which are filled with neurotransmitters. These are these red dots here. And the vesicles undergo a cycle, become closer to the membrane, they undergo fusion, and they release the neurotransmitter that activate postsynaptic receptors which are sitting in the next cell, and this triggers the activation of the postsynaptic cells and brain activity. Now it is thought that changes in the strength in the synaptic transmission, which is called synaptic plasticity, is the basis, the cellular basis for learning and memory. And how fusion occurs? So the basic players are three proteins from the snare family. Two of them are sitting in the plasma membrane. These are syntaxin and SNAP25. They form a complex with another protein, which is sitting on the vesicle, which is called synaptobrevin, or VAMP. This is a very stable complex, which is known as the snare complex. And this complex allows the fusion, and this usually occurs following the influx of calcium through calcium channel, activation of another protein, and initiation of fusion. And this is now how the vesicle fuses and releases its content. Now, there are many other proteins which control the formation of snare complexes, and our hypothesis is that if you have more snare complexes, you will have more secretion or more transmission. If you have less snare complexes formed, you will have less secretion, and this can downregulate transmission, all of them related to synaptic transmission. So changes in the efficacy of synaptic transmission or synaptic plasticity is the main player in learning and memory. Now, it is thought that synaptic plasticity is associated with changes in the expression level and the interaction between these three proteins and many other more proteins. So the objective of our work is to correlate between protein nanometric distribution and synaptic plasticity. In this part of the talk today, I will only describe you the initial goal, describing the nanometric architecture of the snare protein, but this is part of the overall game that we are studying. So the snare proteins on the membrane can either be located at single molecule, they can be found in clusters of one protein or another, they can be found in complexes, three of the protein together. And the idea that we wanted to understand is to learn about the nanoarchitecture of snare protein and clusters using super resolution microscopy, and then to correlate it with the kinetics of snare complex formation using imaging and simulation. And throughout the talk, I will try to connect three angles: the distribution, the kinetic, and the structure. The first challenge is how to reach a single molecule resolution. And we are limited by Abbe's law of diffraction limitation. However, in the last 20 years, many super resolution techniques have started to pop out, I can say. STED, SIM, PALM, STORM, and we have used the D-STORM in the lecture I'm going to present you. D-STORM can take the resolution down to 10 to 20 nanometer. How do you do it? Basically, we separate emission in time. Usually, when you take an image, you take the image of all the fluorophores that you have in your dish, and if they are close together, you'll have a blur, and you won't be able to detect between the images. What we use is specific fluorophore, actually the commercial ones, Psi5 or Alexa 648, or special GFP photoactivatable protein that can switch or photoswitch between bright fluorescent on state and non-fluorescent off state. In that way, what we can create is excitation of few molecules in each frame. And each time, in each frame, you can excite 4 or 5 or 6 molecules. Each one of these spots here represents a single molecule. You can fit a Gaussian with a point strain function and create and get a very accurate definition of the center of each one of these molecules with a 10 to 20 nanometer resolution. And basically, the way we do it is we excite thousands of images. The molecules blink on and off. For example, in this image here, you have 4 molecules that shine, and then you have 4 spots here, another 4 here, another 8 here, and we create thousands of images. We sum them up, the centers of these images, and you create super resolution images by detecting the single molecules in the object. And this is, for example, how a conventional imaging of a microtubule and coated pits will look, or cluttering coated pits. When super resolution, you have a very nice resolution of single microtubules with, and the cluttering is also shown very nicely. So the objective of this initial part is to detect single snare complexes, molecule versus complexes, and to elucidate the internal structure of snare clusters. So the system that we used are PC12 cells, which are a model for a neuroendocrine cell or for neurons. What we do is we sunicate the cell, we rip the upper part of the membrane away, and we stay with the membrane attached to the plasma membrane. This is a very nice preparation since you have all the membrane component on the membrane. You can add antibodies, you can add enzymes, and you can measure both kinetics of different reaction or the detection of single molecules or cluster of molecules. And we then add antibodies, stain them with secondary antibodies, and image the cell. And this image here, it's a bit too much light here, but I think you can still visualize. This is a regular conventional microscopy. You can hardly detect the different organelles here. And this is the storm image of the same, of the same cell. You can clearly see spots here, and now the goal is to say whether these spots are single molecules or clusters of molecules. And for that, we use several algorithms to be able to say whether we have a single molecule or a cluster. First, we, according to the density of the localization or the molecules, we localize the center of mass. And then with an algorithm that look for the nearest neighbors, we search for the 16 neighbors in a radius of 30 nanometer, and I can come back to that later to explain, and detected or defined which molecules here belongs to a cluster and which one belongs to a single molecule on the membrane. And the analysis allows us to differentiate between clusters, which are shown here in red, and single molecules, which are shown here in black. So this is the first step of detecting single molecule versus cluster molecules. When we looked at the distribution of the single molecule, we found several interesting things. First, they are not homogeneously distributed on the membrane. There are areas without any molecules, and most of the single molecules are located very close to the periphery of a cluster. Actually, 80% of the single molecule population resides up to 120 nanometer away from the cluster. And this is completely different from what people describe a membrane like a homogeneous distribution of molecules. So there are clusters, and surrounding them are the single molecule, and this might suggest that these clusters of molecules might act as a hotspot for specific activity in the cell. How does the clusters look like? In previous work using STED, it was suggested that a cluster of a syntaxin molecule, for example, is homogeneously look like a bucket of flowers. However, when we invasion into the cluster with the storm, we found out that the clusters themselves have a gradient. So the molecules in the center of the cluster are very dense, and there is a decreasing gradient of molecules towards the edge of the cluster. So this will be the periphery of the cluster, and this will be the center of the cluster. So there is a very nice gradient almost in all the clusters that we detected. In a way, 60% of the syntaxin molecules are located in clusters. The cluster mean diameter is around 90 nanometer, 93 nanometer, and it has an elliptical shape. And the density of the cluster is about 14 clusters per micrometer square. Not so important, but this is a way to characterize the clusters. We did the same thing for SNAP25, which is a member of the SNARE protein, also located in the membrane. And there are very common feature. SNAP25 also has a gradient. It's also located in also the single molecules are located very close to the cluster themselves. But if we compare between them, there are some changes. So the SNAP-25 clusters are larger, they're a bit more elliptic, and they are less dense on the membrane. But in a way, they have very similar structure like the syntaxin cluster. Interestingly, what we detected is in some mega clusters, we detected several gradients. This, like this one or this one here. This might suggest that the formation of a big cluster is a step of combining small clusters that unite to each other. It could be that these clusters move, or it can be that small single vesicle just invade the distance between the two clusters, and later on they become a big cluster. There is no kinetics here, so this is a snapshot. But also, even in this single snapshot, you can see different types of gradient. This is a single gradient. These are the three gradients, and this one might have a gradient that just fused with the other one. So just to sum up this part of the talk, what I showed you here that there is a highly dense score for these gradients, and this might cause because of protein-protein interaction. The other part of the cluster, the density of the molecules is much less dense, and this might allow interaction of these molecules with other different molecules in the surrounding of the membrane. So the next question that we wanted to understand or to answer is, what is the physiological relevance of protein located at a different part of this cluster? For example, is this molecule can interact with other molecules while these molecules are less active or can less interact? And for that, we wanted to look about the kinetics of snare complex formation. The objectives was to see what is the kinetic of snare complex formation and what is the relation between the positions of the syntaxin molecules at the center or at the edge of the cluster to its reactivity. And just a short introduction about the cluster or about the complexes that this snare can form. So the snare complexes, the two proteins that I mentioned, syntaxin and SNAP-25, can create a binary complex. This binary complex can then interact with another protein, synaptobrevin, which is also found at the membrane, creating what we call the cis snare complexes. And what we wanted to do is to track the formation of these snare complexes on the membrane, and later on try to correlate them with the structural data. We used the same PC12 cell membrane sheet. But at this time, what we did is we incubated the membrane sheet for different time points, allowing snare complex to form. After they form, we fixed the cell, and we wanted to be able to differentiate between single syntaxin molecules and syntaxin molecules which form the snare complex. This was very easily done using the botulinum toxin C, which cleaves only syntaxins which is monomeric or single syntaxin molecules, leaving all the syntaxin which is in the snare complex untouched. This is more or less the same Botox that some of us inject for other reasons. After doing that, we stained for syntaxin, and what we are looking for is an increase in fluorescence, which will represent specifically snare complex formation, or snare complex assembly. And what you can see here in these images is in zero time there is not much fluorescence, and when time goes up to 30, 60 minutes, you can see a lot of fluorescence add up, suggesting that there are more snare complexes formed. What we found out that is the formation, the kinetics of formation is biphasic. There is an initial phase, which is rather fast, within few minutes, which levels off after about 10 minutes. And then there is a second, slower phase of formation of snare complexes. So the complex assembly occurs in two phases. We also did the opposite process, looking for disassembly. And I'm not going to show you the data, but snare complex disassembly also occur in two phases. What we wanted to do now is to build a set of differential rate equations to try to describe the process, a simulation that describes the formation of snare complexes. And the idea was very simple. Let's assume that we have single proteins here, which can form a complex here. We can learn about the kinetics of formation of this complex by just multiplying the K on by the concentration of these proteins minus the K off, multiply the concentration of this complex, a simple differential equation. We had to search for the different rate constant, and since we have several steps, we searched for the proper coefficient based on genetic algorithm. The initial model that we decided to take is a very simple one, a linear formation of snare complexes, A, B, C, and D. And when we tried to fit the data with this model, the fitting was very bad. There was no, we could not really fit the initial phase, and it was rough fitting, but not very good. We tried many other changes using this simple model, but it didn't work. And the solution was to try to use the different distribution of syntaxin on the membrane and to assume that we have two populations of syntaxin. So we assume that we have a population of single molecule syntaxin that can form complexes, and then there is another population of syntaxin which might sit in a cluster, maybe in the big cluster that I showed you before, and can form the complexes in a slower kinetics. Basically, the genetic algorithm searched for us the kinetics. However, this was not enough. So this was the assumption. We have single syntaxin molecule, we have syntaxin sitting in the cluster, but the fitting was not accurate. And what we had to assume is that part of these syntaxin sitting in the clusters are non-reactive. So we have a core which cannot react. We have some molecules in the cluster that can react slowly. Everything is in equilibrium, and we have several syntaxin molecules which are in the membrane. And using this simulation, we got a very nice fitting for the two-phase formation of snare complexes, the fast phase and the slow phase. So if we try to interpret the data, we suggest that the fast kinetics phase of formation of snare represented by the single syntaxin molecule pool, and which are accessible for interaction. The slow kinetic phase are the clusters, and we have an inactive molecule in the center of the cluster. They cannot form a cluster. And then we have a semi-active molecules at the rim of the cluster accessible for protein. So can be correlated with the D-storm analysis. And this is very nice. And this is shown in this slide. We assume that the single syntaxin molecule that the model suggests can form a fast complexes are these black points surrounding the clusters. Now the clusters themselves can be divided into two parts. We have the molecules at the rim of the cluster that can form snare complexes in a slow process, and then we have the non-reactive syntaxin, which might be represented here by the core of the cluster. So this vesicle that cannot form clusters. So the results suggest for the first time a link between the inner organization of protein in a cluster and their kinetics properties by the formation of snare complexes. What we are doing now is we are trying to simulate a 2D simulation describing the movement of the protein on the membrane and the formation of clusters. I'll try to show you briefly the simulation, if it works. So you can see here, these are the clusters in the RAF. These are single molecules. What we can do now is easily add another protein, for example. Let's see if it works. I have some resolution problem here. But I think the idea is clear. We can add more proteins, and we can change the reactivity between the protein. And in that way we have a nice 2D simulation that we can now extract new parameters that we could not do before. So just to summarize, I showed you that syntaxin and SNAP25 are distributed between clusters and single molecule pools. We observed differences in cluster size, shape, and density. We could invade and describe the internal density of the clusters and show that there is a gradient. And we can really detect single molecules versus clusters. And I think the biggest advantage is the combination of super resolution microscopy imaging, in this term, D-storm, with cell biology and with modeling in order to be able to tell much more about complexes of proteins in the cellular environment. And I just want to thank the people who did the work. This is a collaboration between Markus Sauer, who invented the D-storm, and Chemi Gutman and Eti Nachriel from the biochemistry department. And all the work that I presented was done by this talented PhD student, Dana Bar-On, who is now finishing her PhD, and Amit Alon is now continuing her work using neurons. Thank you.
Questions?
Okay, I have a question. You said that you need to fluorescently label the proteins to do imaging. And how do you do it in a living cell, and does it not interfere with the normal function of the proteins?
That's a very good question, I think, general to all of what we are doing. So the work that I showed you here was with endogenous protein using antibodies. There are several options in using a photoactivatable protein that you can overexpress. There are some promoters which are less active, like the CryptCMV, that you can express in low level, but you never know whether the expression itself will change the physiology of the cell. So that's, but you can use these molecules also in live, although I think this is, only the beginning of the way in D-storm or storm.
Is there a chemical significance to clusters, or is just self-assembly of...
It was suggested that there are interaction between the different molecules which create this clustering or oligomerization. But I'm not sure what you're talking about, chemical covalently bonds or...
Because it seems to me that in an arbitrary way, just by measuring distances, you decide which molecule belongs to a cluster and which does not. There's no different fluorescence on other physical...
No. At the moment, we couldn't detect any differences in fluorescent or number of blinking between molecules that we defined as clusters. We had to play with it, but we might have missed. But I think if you look by eyes, it seemed very nicely that the cluster that we defined are real clusters. But this is always a...
How much time does it take to take a picture with the D-storms to get a...
I think if you're using the Alexa, which are very bright, you can take a 10 millisecond image. You need something like 10,000 images or 5,000 images, so it's a minute or so.
So you cannot really learn about dynamics of...
There have been one or two publications looking at dynamics. When you do the dynamics, you lose, I guess, quite a lot. But if you don't need to see all the molecules, that might be enough. This is, at the moment, a limitation, I think, to move from fixed to live cells. But this might be, I think, challenged by very fast cameras, which can take 1,000 images per second if you have a bright fluorophores.
Okay, so thank you all again.